Welcome to MassBay's Biotechnology Immunology class of the spring semester 2021. This semester, we decided to study the effect curcumin has on the rescue of splicing defects and oxidative stress caused by paraquat in human fibroblast cells. Our goal of this experiment was to test curcumin's ability to save human cells from possible harmful defects caused by toxins and popular herbicides. We also wanted to test curcumin with genetically defective human cells to see if curcumin would be able to help restore genetic defects. So what is curcumin? Curcumin is a chemical compound found in the common spice turmeric. Turmeric has gained popularity for its anti-inflammatory and antioxidant properties. Recently, curcumin has been under the microscope in studies regarding its ability to help modify gene expression. We needed a toxic chemical that would induce cell damage to test curcumin's saving abilities. We chose paraquat because it's a common herbicide used in the U.S., so our experiment had relevance. We also wanted to choose paraquat because it's a known toxin, so we would expect our cells to be affected by it. Studies have shown there's a possible link between paraquat and neurodegenerative diseases such as Parkinson's and Alzheimer's. This increases the importance of research needed to discover potential treatments that could counteract paraquat's harmful tendencies. Studying the way our genes can be turned on and off is a new field called epigenetics. Epigenetics has become one of the most exciting fields in the scientific world, and to help expand our knowledge, we decided to perform our own epigenetic experiment with curcumin. Aside from testing curcumin against toxins in our environment, we wanted to take our studies a step further by testing cells affected by gene defects to see if curcumin was able to help mend the genetic damage. To test this epigenetic factor of curcumin, we chose to also study SMA cells. SMA, or spinal muscular atrophy, is a genetic disorder where a person has a mutated gene. This mutated gene prevents the body from creating very important proteins that help with the vital communication between the spinal cord and the muscle cells. Our goal was to see curcumin's epigenetic ability would help mend this genetic mutation and help produce more of these very important proteins. To start this experiment, we housed our two cell lines, healthy cells and SMA cells, in an incubator in the lab. We performed cell culture, and we treated both cell lines with varied amounts of curcumin and paraquat to test the different potencies. We then extracted genetic code that we were able to purify. We took the purified genetic code and produced DNA for each cell line. We were able to process this DNA to show what genes were being expressed in our cells. Our results show that curcumin has a positive rescue effect in healthy cells that were exposed to the toxin paraquat. These results also show that curcumin has a positive effect on SMA cells in mending the mutated gene. Mending the mutated gene on SMA cells means these cells now produce more of the very important protein needed for muscle movement. This proves that curcumin can in fact modify gene expression. We can conclude through our research that curcumin can aid in genetic regulation as well as helping cells prevent damage caused by toxins in the environment. Our experiment shows us the possibilities curcumin can provide by being an easily obtainable preventative treatment to potentially harmful chemicals we find in our everyday environments.